welcome back to my channel, and in today's video I'll be discussing DNA polymerase which are found in prokaryotes. Okay, prokaryotes, specifically if I say bacteria, E. coli, they usually divide two daughter cells. They divide by a fission and produce two daughter cells. The prerequisite of the cell division is copying, or I can say replication of the genome. That is, before the cell division, in order for the cell division to take place, before the formation of the two daughter cell from a parent, DNA replication needs to be done. This DNA replication in prokaryotes is done by an enzyme which is termed as DNA polymerase. DNA polymerase is involved in the replication, copying, I can say, of the genome of the prokaryote, of the bacteria. Okay, the next question here comes. During this replication of the genome in a prokaryote, is there only one enzyme which is doing that particular job, or there are a number of enzymes coming together to replicate the genome? So, DNA polymerase in prokaryotes, they are found in 5 different types. Okay, DNA polymerase 1, 2, 3, 4 and 5. Okay, there are 5 different types of DNA polymerases in prokaryotes which are involved in the replication of the genome. The next question here arises is, are they all these 5 types of DNA polymerases, are they doing the same job? Are they doing the same function, or they have divided the labor of replication among themselves? So in this video, I'll be discussing the functions of the various DNA polymerases which are found in prokaryotes, which are very necessary for the replication of the genome, which is a prerequisite of cell division to take place. Let us study the different functions of the DNA polymerases which are found in the prokaryotes. The DNA polymerase 1 is involved in the DNA repair mechanism. Okay, if the DNA requires any kind of repair, DNA polymerase 1 will perform that function. Okay, in order to understand the next step of the function of the DNA polymerase, let us briefly discuss the replication which is happening in the prokaryotes. Okay, the replication of the DNA. Okay, DNA is double stranded, right? It is double stranded. Okay, they separate, and both of them, they are the template strands. Okay, that is, the replication of these both strands will occur simultaneously. Okay, and in this replication, one strand is synthesized in a continuous manner. It is replicated in a continuous manner. Okay, the replication of this strand is continuous. On the other hand, the replication of the other strand is discontinuous. It occurs in fragments. Okay, the strand which is undergoing a continuous replication is termed as the leading strand, and the alternatively the strand which is going discontinuous replication is termed as the lagging strand. Okay, these fragments which are generated during the replication of the genome, they are termed as Okazaki fragments. Okay, now DNA polymerase is such an enzyme which itself, if I say that this is a DNA template and I have to in the cell it has to be replicated, it has to be replicated, so DNA polymerase cannot go and start the replication on its own. Okay, it requires a primer. Okay, it requires a primer, an RNA primer. If I talk specifically, this RNA primer provides the 3' OH. Okay, it provides the 3' OH, to which this DNA polymerase now can go and add a nucleotide here, can start adding the nucleotide here, can start the polymerizing reaction. Okay, so these DNA polymerases, particular enzyme, it does not goes and starts replication on its own. It requires a 3' OH, which is usually provided by a RNA primer. So RNA primer will be present at here. Okay, one side. Whereas in case of the Okazaki fragment, each fragment is started by an RNA primer followed by the polymerization reaction by the DNA polymerase. Each fragment is started with an RNA primer and followed by the adding of the nucleotide by the DNA polymerase. Okay, similar goes here. Okay, so as to make this strand continues, so as to make this particular strand continues from its discontinuous part, what happens? These RNA primers needed to be removed. First step would be removal of the RNA primers, right? We have to remove these RNA primers. The next step will be sealing them, okay? That is adding nucleotides between these gaps. Okay, this removal of the RNA, replacing it with the DNA, it is usually done by DNA polymerase 1. Okay, it connects the Okazaki fragments by replacing RNA primer with the DNA, leading to the formation of a continuous strand. And the last is sealed by another enzyme which is termed as DNA ligase. Okay, that was the function of DNA polymerase 1. It is playing a very major role in DNA replication. Okay, DNA polymerase 2, it is also taking part in the DNA replication. DNA polymerase 3, it is the primary enzyme. What do you mean by primary enzyme? By primary enzyme here, it means that this is the main enzyme which is acting the function, which is playing the role, which is involved in DNA replication in prokaryotes. Okay, it is a primary enzyme. So what happens after there is laying of the RNA primer, which polymerase is coming and extending, that is DNA polymerase 3. Okay, DNA polymerase 3 is responsible for adding the nucleotides to the 3' OH, which is provided by the RNA primer. Okay, in other words, I can say it is a component of replisome. It is responsible for the replication, the main enzyme, the primary enzyme. Okay, now next two enzymes, that is DNA polymerase 4 and DNA polymerase 5. DNA polymerase 4, it is an error prone DNA polymerase. Okay, it is an error prone DNA polymerase. It is an enzyme which is error prone. During the performing the function of polymerization, it can result into some errors. What errors am I talking about here? We all know the complementarity concept. A pairs with T, C pairs with G. Okay, DNA polymerase 3 is a high fidelity enzyme. What do I mean by high fidelity? That is the complementarity, that is A always pairs with T and C always pairs with G, and the error rate done by DNA polymerase 3 is very, very low. That is one of the characteristics of it. It is a primary enzyme. Whereas in case of DNA polymerase 4, it is an error prone DNA polymerase. That is, at some point it can add A against G. If I can say it for some time it can forget the complementarity rule. Okay, and as a result of this error prone, as a result of this mispairing created by this enzyme, it is involved in the synthesis of the mutations, that is, which are usually untargeted. That is, it is involved in untargeted mutagenesis. It is involved in the untargeted mutagenesis. Okay. Next enzyme, that is DNA polymerase 5th. This is the last enzyme which I'll be discussing. And the major function of this enzyme is trans-lesion DNA synthesis. Okay, this particular enzyme is involved in a phenomenon which is termed as trans-lesion DNA synthesis. Before going into this, what does it mean? Let us study another characteristic of DNA polymerase 3. Okay, whenever this enzyme, DNA polymerase 3, it adds a nucleotide which is not complementing the template strand. What does it do? Suppose While performing its function, it is replicating the template strand, it is doing its function, and a point comes where it adds G against the template strand, which is A. This is a template strand, it is having A, it has added G. What does it will do? DNA polymerase 3 will stall, it will halt, it won't go further. Okay, it will stop its function, it will take a pause. After taking a pause, DNA polymerase 3 is having a 3' to 5' exonuclease activity. Okay, it will have 3' to 5'. It is having 3' to 5' exonuclease activity, which is also termed as a proofreading activity. It will remove this G and add a T. After taking a pause, it will remove this G via its proofreading activity and add T and continues this replication. But DNA polymerase 5 doesn't do it. It is a translesion DNA synthesis type of DNA polymerase. It would not stall, it would not pause, it would not halt. What it will do? It will skip it. It goes beyond it. It doesn't care if it is mispairing or what it is doing. What it does, it skips this and continues its work. There is no pause, there is no halting, there is no proofreading activity. No, what it does, it skips this point where mutation has taken place, or I can say mispairing has taken place. Okay, it is a translesion DNA synthesis enzyme. That was all about the functions and the types of the DNA polymerases which are found in the prokaryotes. Thank you so much for watching my channel. Please like, subscribe and share.